Hello, everybody. We'll be starting the second BTO conference session in 1 minute now. Hello, everyone. Good afternoon, and welcome to the second session of BTO's virtual conference. We're really sorry that this year the pandemic means that we're unable to meet you all in person. However, it is exciting that at the moment we have almost 250 participants connected to the Zoom webinar and doubtless more watching on our Facebook and YouTube live streams, which is far more than we could have probably hoped to have fitted into our usual in-person conference. I'm Sam Franks. I'll be your chair today for the session, and I'm a senior research ecologist in the wetland and marine team based in our Thetford office, and I'll be your host. First off, if you missed yesterday evening's fantastic session on tracking, then you can head over to our YouTube channel to catch up, or you can find it linked from the conference page on our website. This afternoon, we've got 3 fantastic talks lined up about waders, which is a topic very dear to my heart. First off, though, a bit of housekeeping. After each talk, we'll have a short time for questions. We have disabled the chat function in the Zoom webinar, so please use the Q&A button at the bottom of the screen. Try and use this only for questions about talks. We have several other staff members who are monitoring the session, so any technical issues that you might encounter should hopefully get sorted quite quickly. But if you must ask because it's not sorted after a short time, then please do use the Q&A for that. You can like or thumbs up any questions that you already see, which will bump these closer to the top of our list during question time. Also, there will be a staff member who are monitoring the YouTube and the Facebook live stream, so we'll try and tackle a few questions from those media as well. Any questions that you have of a more general nature, please save those for the AGM on Saturday afternoon. We have several more days of talks and panel discussions lined up, so please do tune in over the course of this week. They're coming at a range of different times to suit different people's schedules, so please do tune in if you have a chance. All of this that we're presenting now is possible thanks to the support of BTO supporters, and so very much thank you to those of you who are here today. Income from membership and donations makes up half of the BTO's income, and without that support, BTO wouldn't be what it is today. It's a tough time for many right now, and for charities like ours, income is suffering and the future is quite uncertain as a result of COVID. Many of you already support us in a whole variety of ways as members, volunteers, or by donating, and we're really grateful for whatever kind of support that you are able to provide. If you are in a position right now to make a donation at this time to support our work, then we'd be extremely grateful. And you can do that via the conference link that is being shown on our screen now, www.bto.org/support. Also, if you enjoy today's talks and feel like BTO is an organization that you'd like to support, then please consider joining us if you aren't a member already. Our members really are our lifeblood, and it's through membership support that we can inspire and educate people and together advance our collective understanding of birds through research. So we'll get going now with our first talk, which will be presented by BTO staff member Katharine Bowgen, who is a research ecologist in our Wales office based in Bangor, and she'll be telling us about how GPS tracking data can reveal details about curlew breeding behavior without the need to ever visit a nest. Over to you, Katharine.
Thank you very much. Hello, everyone. My name's Katharine Bowgen. I'm a research ecologist at the British Trust for Ornithology, and as Sam mentioned, I'm in the Cymru office based in Bangor. I'm also part of the wetland and marine team. Some of you might have remembered that last year at the conference, I gave you a talk on the tracking work that we've been doing up in the North Wales uplands and lowlands. But this year, I'd like to give you an idea of what we can do with those tracking movements just beyond being able to understand where they're going. So we have this idea of why we should track curlew, and as people know, and it's unsurprising to a group such as yourselves, we have a huge range contraction and decline in population of curlews across Europe, and particularly in the UK and Wales. If we look in particular at the last sort of years, as you can see in the bottom left of my plot, we can see there's a serious decline going on, and we want to understand a bit more about what's happening, why this is happening. We can do this through general movement analysis, but also we can understand a bit more about what is going on with their breeding stages, which is one aspect of the life cycle that is important to the birds up in North Wales. I've got this lovely image that I have shown before just to show you the importance of curlew around the country, within Wales as a country, that is, basically showing all the different personal connections people have with the different names that are coming up for curlews. Gulfinia is the main name that is given, but every single part of the country has a different name for them, whether it's from Beggy Big here, which is sort of Peggy Big Nose, through to names that are more to do with mountain whistlers. We understand they're important to everyone, and so we want to find out a bit more of this evocative species. So trying to go into why I'm taking this a bit further. So identification of species that are breeding who are really cryptic, such as curlew, is really tricky. We don't actually understand a lot about what's going on at their breeding stages, whether this is when they start incubating, whether they actually have chicks, whether they're successful. And being able to track them is very tricky. As you can see from the top right image, I'm not sure how many of you can actually spot where this curlew is, sitting right there in the green circle oval. And we can see that it's really tricky to pick up where they are. And you can walk through a field and actually only a few birds in that field will alarm call. And so if you're trying to do studies of figuring out how many birds are there, you realize you actually might be short of numbers. And we also know that they can travel huge distances from the work that we've been carrying out. And so we might end up double counting some of them. Trying to understand more about their breeding cycles do mean that we do make them more prone to disturbance, and there's potential for abandonment. And so we're trying to understand more about what's going on through these tracking methods, which are easy to put on. They're very safe for them. Their tags in particular drop off really quickly because they're pushed out by the feathers. And it allows us to actually pick up birds that we can figure out what's going on, but also when we're trying to pick up what's happening with their chicks as well. So we can't tag the chicks. And as many people know, these chicks are up and running really, really quickly. Some of you might have watched Curlew Country's live webcams that have been happening for the past few years. And the images across the bottom are 3 screenshots from the day that the chicks hatch. From the left, we have them just pipping out of the eggs through to sort of warming up and fluffing up a bit, to the point on the right where we can see a chick's head just poking out from underneath the adult, and they were often gone before midday. It's very quick. It's very hard to actually monitor what goes on beyond the nesting stage, even if we've been lucky enough to get a camera. So we've been lucky enough that we've been funded through NRW and other sources to be able to put tags on them. These are GPS tags. They allow us to be able to track what the birds are doing over the course of the breeding cycle. And they are small little devices, very lightweight, small percentage of their body mass. And we put these on the birds, we attach them via glue, and then they are managed through pushing off from the feathers that have been trimmed down. They sort of fall off by the end of the season, either through preening or natural feather loss. And we're able to also monitor the birds from the colourings that we've put on to identify as well from distances what's happening with them. Now, we've talked about this in many studies before, and last night they had talks that were going on about sort of tracking and monitoring. What I'm going to talk to you is a bit more technical. Now, I'm not going to go into this too deeply, but for those of you who are interested, we're trying to figure out where the nesting sites might be. Now, there's a couple of packages that are available within statistical software, in particular a new one called Recurse, that allows us the opportunity to figure out hotspots of interest. And you can see in the plot on the right, all of the relocations of 2 birds in red and in blue. They picked out the areas of interest in the darker, more bolder shapes. We know quite where the clusters of interest are them. And this package was developed to identify roosting sites, foraging hotspots, but also nesting locations. And it's based off the time spent in an area rather than just coordinate clusters. And this allows us to truly understand where the birds might be spending time, in particular when they're nesting. We know that they will be spending incubation time in an area, so they'll be fixed on this particular area for a decent amount of time. But to try and give this in a bit more of a context and a story, we have a curlew that we called Vincent. His name for Vincent is to do with the local farmer around, but he's really quite characteristic in our minds because we actually know what happened to him. He was one who was well monitored by some local wildlife trust members, also monitored by some RSPB staff and the farmer himself. And we know that he was a male who hatched 4 eggs in 2018. Sadly, we also know the date that he lost them. This is not part of the story for any other reason than just that we know when the start and end date of his eggs were, when they hatched, and we know roughly when he lost them on time of day. Now, you can tell from the cluster of points on this Google Earth from the first images, we know where his nest site was. It was really nice and clustered around this field. We also know that when he started to hatch the chicks, that he spread out, but on the day that he lost them, he suddenly moved his movements all the way around, and we know that he spread them out to the point that we can zoom right out and see that it actually ended up heading down towards the estuary. This is fantastic for us, as much as it's a sad occasion, in the fact that it allows us to be able to pinpoint roughly when we know the changes in his breeding status happened. So when they went from incubation to chick guarding through to loss. And we kind of thought, okay, let's see if we can pick out where his nest site was from the days that we know he was incubating. Now, this new package picked out these really nice hotspots of interest. So you can see, don't worry about the rest of the graph, but you can see how the red blobs in the top, they show us right where his nest site was. And then we can see in week 23 that he spread out a bit as he had small chicks moving around. Whereas by week 24, he was spread out much more. He wasn't focused on the same areas again because we know he'd lost his chicks. They're still going backwards and forwards. And we thought, okay, that's great. We can actually pick out where his nest site is. But we also discovered these plots as well. Now, it just happened to be that within the recommended process for using this package, they said, oh, why don't you make a histogram of the frequencies? And they're a bit tricky to explain, but essentially they show you how often a bird was using an area. So the more to the right of the plot, that means that a bird was revisiting an area over and over again. And then how high the plot was how many times they were revisiting multiple times. And we noticed that in the top row, when it was incubating, there was this nice hump over to the right. This is because it was using an area and it was picking up this area and going, yes, this is where my incubation site is. I'm visiting this for a long time, lots of times. Week 23, when they had chicks, shifted all the way over to the left. And we suddenly found that we had this more sloping plot, which allowed us to understand that, oh, so this is what happens when they got chicks. And it makes sense if you think behaviorally, chicks can't move very fast, but they are movable. So they're moving around, but they're not going to random locations. They're slowly working their way through the areas of their habitat. Week 24 is when we know that we started to lose the chicks, that they were lost. And you can see that the diagonal sloping plot starts to shrink upwards towards week 25 when we know he had no chicks. And the very tall bar on the left, that shows us that the birds were basically visiting a site over and over and over again. We're visiting new sites, not over and over again. They were visiting sites uniquely, and they weren't following back to the same site as often. Taking those days, we were like, okay, so we actually know what's happening. Can we pinpoint this down to the hour? And these are all the relocations we gathered for the 3rd of June and the 4th of June. And by doing my analysis and moving it as a window over all the different points and days, I was able to find that the normal incubation pattern on 3rd of June split up on the 4th of June. And you can see that the dark colours are early in the morning, working their way through midday in a blue to late at night in a yellow. And we can see that actually this bird, instead of staying overnight in the early hours or the late hours in the same place, had shifted it south. And this is when he started chick guarding, which means that we actually know that the chicks were up and moving that morning, which is really cool. It also shows us which is the point of failure. So on the 12th of June, we had lots of movements with young chicks sort of pottering around the area. On the day of chick loss, you can see that he's still kind of around the same area, but he's shifted over dramatically. And the yellow dots show that he spent his night further away from that site. By the 14th of June, he still had a few visits back to the same area where the chicks had been, but very different pattern and wasn't moving around. This meant that by picking out just from the relocations without observing in the field at all, we could have worked out what was happening to the bird. And we wanted to apply this to the other birds. So I mentioned we had 26 other tags out, and we got 23 tags worth of data back. So we decided to try this, and we found it worked. This is a bird who was nesting in the eastern uplands of North Wales. And we can see on the left, we could pick out an incubation pattern. We could also pick out a slightly more sloping pattern. Now, you can tell this isn't quite as strong as the other one. And of course, there are slight differences between how birds use the landscape and how the relocations come back due to satellites and all the other technical parts. But this bird was later on pointed out to us to have had chicks. So we went back and looked, and we found that this was the point at which it had chicks. So that's great. We were also able to find out an incubation peak for another bird. And again, individually, this bird was quite different from the others, but still had a peak. And then failure, dropped right down, bird headed off, and we didn't get any more of these strong patterns again. And finally, I should point out, we could also figure out which were the non-breeders. And actually, this is almost more important in some cases for trying to pick up breeding populations. Working out which birds were around and giving territorial signs, but never incubated, is actually really important for us to be able to gather how many birds are likely to have had chicks opposed to the others. So I want you to fully understand this plot because I realize it's a bit complicated. But the idea was that for every single one of our tagged birds, we were able to figure out whether they incubated, whether they had chicks, and whether they failed. What you might notice is that the columns to the right, green is when we know something happened. So the first column of greens is all the birds that incubated. So there were only 6 birds that didn't incubate at all that were breeders. All the others had at least one go at incubation. A high number of them failed from looking at the plots and the movement patterns. But we had another bunch who incubated for a second time, and we never would have known they'd incubated for a second time without being able to analyze this data quite as we had. We would have known they're going back to the same areas, but this allows us to truly know that they tried again. And in one case, a bird in 2019 was incubating for a 3rd time. And you might notice that a number of these don't have any information further to the right. And this is because the birds managed to get the tags off before the end of the breeding season, which means that we know that they were incubating the last time the tag was on. Across each of the 3 years that we did this, we know that we had at least one bird that managed to get birds to chicks to a chick guarding stage. They hatched and they were out, which is great. We know that were successes. And we also know from observations later on in the summer that young chicks and young adult birds were seen, and we know that they have been able to make this to stage. And so this sort of analysis helps us take the movement analysis a stage further. I thought I'd give you a bit more of a straightforward table, so you understand that in each of the years, the number of birds that were tagged that we could analyse, which was 23. In 2019, 12 of those 15 birds made an attempt at incubation. So sort of well over 80% of each of these were making tries, and we had 7 retried in 2019 as well. We did sadly have 13 failures, which is a shame, and we know that this is a major aspect of what might be driving some of the populations if they're not being successful. But the fact that we know that they reattempted, they incubated again, gives us hope that they might be able to work through. One thing I should point out is that this is not the end for Vincent. We actually caught Vincent again in 2019, and we found him incubating. You can see he's back in the same sort of area, if you kind of remember what the picture looked like before. And we can see that he was incubating. Unfortunately, he figured out how to get his tag off, or he was just really, as in he was really good at preening. We only got 34 days of data, and he was still incubating when the tag came off. So we're hopeful that he was able to make it through because there were better provisions in place for his habitat. One other use of this analysis that we had always hoped we'd be able to work out right back before I even found out we could identify between different breeding stages was actually figuring out if we had any pairs. So we actually had both males and females that were tagged, and we understood that there were some slight differences in behaviours. In particular, we know that the males do the chick guarding, but we managed to figure out that 2 of the birds that we had tagged were a pair. These circles on the plot, the green and the red, that overlap strongly and pretty much 90% of overlap, they were 2 individuals that I pulled out separate hotspots of nest site locations, and they overlapped right on top of each other. Both of those birds were a pair. Those birds were a pair. And they also re-nested within the same set of fields, very close to each other. And when we look at their individual movements, of which the males in the red, the females in the yellow, we can actually see they were using very similar habitats around this time. So the main block of colour is where the nest sites were, of course. But we see that though the female went a bit further out west and the male went a bit more northeast, they were still using the same habitats, which is really interesting. And that hopes that in future we'll get more pairs and we'll be able to understand quite how they shift and share their incubation timings and schedules. What I should go on now to talk is our future plans. So hopefully I haven't blinded you with all the science, but I wanted to show that we can actually take this further than just finding out where they go and where they step. We can put this into use that will be able to understand more about the breeding seasons and what's going on. I'm currently writing this up as a publication, so I'd be grateful if not too many tweets were made about this on Twitter if you're doing it that way. Hopefully it'll be sent off to a journal soon. And we hope to expand this analysis into other populations of breeding birds, whether it's within the BTO's own work programs or with other colleagues that have shown interest in my analysis. We have more curlews in Wales and potentially East Anglia that Harry's going to talk about. Potentially these are areas that we could include this analysis on, but also some of the gulls in Scotland and potentially raptors. It'd be really interesting to understand if we can pick out particular patterns for these birds to allow us non-invasive ways of understanding whether they've been successful in their breeding or not. The final thing, which is more statsy, is I would like to automate this. So I have to do this manually. As you could imagine from looking at some of my plots, the actual difference between when they go from incubation to chick guarding, it's not standard. It's really obvious when you've been looking at 90 days worth of a bird's data, what it's doing. But we would like to be able to automate this and work out when it shifts so that we can just run it through and get really quick data back to be able to take it forwards to actually analyse in the field and focus in on birds that are breeding, who've been successful, to maybe follow them a bit further. The final thing I would like to point out is that even though we were not able to get out into the field in 2020 due to COVID, we were given these lovely videos, of which I've just taken a screenshot of curlew chicks that had made it to the running about stage happily in a farmer's field in these areas where I've been following the birds. And we know that they have been successful this year, and fingers crossed we'll be able to get out next year and follow along with what they're doing. Thank you to many people who've been involved in this work, whether it's NRW who've been funding us, and also RSPB who helped with some of the initial collaborations, and all of the various different field workers, some of the students who've helped bring the project to life, and all the farmers and stakeholders. Thank you very much. If anyone's got any questions, I hope I can answer them.
Thanks very much, Katharine, for a really brilliant talk. It's fascinating to see how statistics can help identify where curlews are nesting if you analyse the GPS data appropriately. So we've had a few questions come in the chat now. One that a lot of people are interested in, asked by Sue Perkis, is she'd like to know how big a problem is silage cutting for curlew in Wales?
So in Wales, it's a bit of a problem in some areas. We don't actually know the full extent of how much it might be. We're hoping that with future projects that we might be able to work out quite how much silage is impacting on birds. But one thing I must say is that when we have been working in our areas, we've been able to coordinate with the farmers and the stakeholders who are involved. And if they know that they've got curlew on their land, they actually are very accommodating with being able to change what they're doing. The second bird that I showed that we'd worked out that it was chick guarding, the chicks were actually picked up before we had all the data back. Someone had noticed that there were some chicks in the field, and in coordination with the farmer, he held off for 2 weeks for his silage mowing. And then when he did start mowing, people were observing from the road and they watched the adult shuffle his chicks across the field that was being very slowly mowed into a new field. And I could follow this on the tracks afterwards. And we know that that extra 2 weeks meant those chicks were a little bit taller, less likely to just duck and cover, but more likely to move. So hopefully we'll be able to work out a way, but it does require monitoring and talking, but everyone's very accommodating.
Thanks, Katharine. That's really encouraging to know that there are farmers out there who are willing to change their own schedules to accommodate curlew in that way. We've had another question from Stephen Chapman, who would like to know how old curlew are at first breeding and what their normal life expectancy is.
So curlew tend to start breeding within the first couple of years. So we don't think that they start breeding within their first calendar year of life, but definitely by their third year they will have. And then how long they live is a little bit of a mystery in some cases. We know some birds have made it up to 30 years worth of age. Of the ones that are caught and who are monitored through BTO ringing schemes, we know that they will make it into their teens. But a lot of the birds that we catch are often on wintering sites, which means that they're not actually UK breeders. They're more likely to have come from Scandinavian populations. So the hope is that with more of the monitoring of our own breeding population, we'll understand quite how old they are. But we do know that we have at least some 30-year-old curlews out there.
It's crazy to think that curlew can make it to that age. We've got another question here. So this person is wondering how you're catching the adults, and he'd like to know, because he has some, he's generally interested because he has some curlew local to him on a site in Cumbria.
Fantastic. So great that you have some nearby to you. So we've got one method of catching we're using in North Wales, which is cannon netting, and it's cannon netting for an individual. So people might have heard of cannon netting being used on large flocks of wading birds and on ducks and geese. What we are doing is we are putting a dummy bird out, sort of a model of a curlew. We're playing curlew territorial calls in an area where we know there is breeding males and other females around. And then we are firing a very small net. We have a couple of people with special licenses to catch curlew, and we're catching them that way. But importantly, we're catching them before they start breeding. So we're catching them whilst they're first on the breeding grounds and they're first interested in being territorial, but before there's any chance they might be incubating. So this allows us to be able to put the tags on and get some pre-breeding movements, but also know we're not interfering. There are other methods of catching. Some people catch them on the nest and tag them that way, but it all depends on what your population are doing and what expertise you have around. The cannon netting we're doing works very nicely for a small number of birds.
Great, thank you, Catherine. Got another question here from George, who wants to know whether you can use the data that you're collecting from the GPS tags for nest protection.
Fingers crossed, and that's the plan. So what we do is we have downloading remote base stations. So there's several types of tags out there, and the tags that we have that we're using on the curlew download to a local base station. Someone goes out and they download the data. We had some students going out and downloading the base station data for us last year, along with some other colleagues, and they were able to get data in every few days if they were out that way, or at least once a week. The sooner we can get it in, the more we can analyze the data. And even before I get through all of my long analysis, we can understand what rough movements they're doing. And we can potentially say to a landowner, we think your curlew are breeding. Please be aware of what's going on. And I know other schemes are doing the same thing where they're trying to monitor on a short-term basis. It all depends on how quickly you are able to get out and download the data. But hopefully with the automation of my new process, which fingers crossed we'll be able to do, we would be able to just bung all the data through and someone would get it out just to know whether they are incubating or not.
Great, thank you. We have a question from Mark Wilson, who I'm guessing is our very own Mark Wilson, who says, wants to know if you've thought about using machine learning in the same kind of way that's used for training acoustic data classifiers to help to automate the classification of these GPS data.
Machine learning would be ideal. Unfortunately, with only 23 birds at the moment, and with only 3 of them having shown any signs of chick guarding, we don't have enough to train a machine learning piece of software yet. The hope is that if I can get, well, one, we will get more curlew from this, and also if we can apply it to other curlew around, and potentially some colleagues in Europe have expressed an interest when I've talked about this to them. If we get a large enough data set, we can do machine learning, but at the moment, with only 23 birds and only 3 of them having shown any chick guarding, it's not enough to really train a machine learning. Machine learning requires both the data set to train them on, but also a testing data set, and we don't have enough yet, but hopefully in the future.
Great, thanks. We've got a question from Owen Selly, who wants to know the frequency of GPS fixes and whether you could use the GPS data to work out how often curlew are leaving a nest like you can with temperature loggers, for example, to understand their reaction to predators.
So the loggers that, the temperature loggers that people use are fantastic from being able to know whether there is a hot body sat on top of them like a curlew that's incubating. Our tags take a GPS fix every 15 minutes. Now, the reason they're every 15 minutes, not shorter than that, is because we're trying to prolong the length of the battery life of the tag. Some of our tags have lasted all the way through to August from April, and that's by just using the 15 minutes. They do have a small solar panel on them, so they are able to recharge. Ideally, to be able to know what responses to immediate disturbance, we would need a less than 5-minute interval to be done, but we wouldn't be able to have the tag on long enough for getting enough data from that, so we've not done it. There are hopes that there might be some tags that are able to have dual cycles, so we can have a very fast, if the bird hasn't moved from an area for a while, we can ask it to take minute worth of data, but at the moment it's on 15, so whilst we'll be able to work out rough incubation schedules, we can't get it quite as close yet until, as with all things, we need better battery life on lots of these.
Thanks. And we'll do one question from our Facebook live stream. Apologies to those who I haven't been able to get to. Ronan O'Cara wants to know if there's a consistency in the type of failures you're getting, so whether they're from predation or disturbance from agricultural activity, for example, or are there a whole range of factors, do you think?
For our birds, I must admit, we don't actually know what the causes of failure are in the majority of cases. Vincent was an unusual case, which is why we were able to actually use him in the way we have. But because we are doing this with a non-invasive method, we do not have people out all of the time. One thing I have noticed, though, is quite a lot of failures have been in the early hours of the morning, and that potentially could be related to crepuscular activity of mammals. Now, this is a big speculation, please do not take this, but we are potentially hoping that with further understanding from cameras, so people who've got cameras on the nest as well as a tracking logger, we would be able to associate them, but at the moment we can't tell, sadly.
Thanks very much, Katharine, for answering all of those questions and for a brilliant talk. We're going to have to move on now. I'm afraid I won't be able to get to everybody's questions. Apologies there. So we're going to move on to the second talk of this session now, which will be presented by Harry Ewing, who is a PhD student at the UEA, co-supervised by the BTO and doing fantastic work on breeding curlew, which are local to our BTO office in Thetford. Over to you, Harry.
Thanks very much, Sam. Hi, everyone. My name's Harry. I'm a PhD student at UEA, and today I'm going to share with you all the work I've been getting up to over the first half of my PhD, which is all about the breeding ecology and conservation of breeding waders, with a particular focus on the curlew. So, as I'm sure most of you know, breeding waders are declining all over the UK. And BBS trends suggest that over the last 20 to 25 years, we've lost around 24% of our breeding oystercatcher, 43% of our breeding lapwing, and 42% of our breeding redshank in the UK. Now, we think these declines are due to unsustainably low rates of breeding productivity, caused mainly by high rates of nest and chick predation by these guys, generalist mesopredators. Now, in the UK, we have really quite high densities of these predators. So if we take foxes, for example, this really nice study by Roos et al. Showed that out of all of these different European countries down the bottom here, the UK hosts the second highest density of foxes, and England hosts the third highest density of foxes. It's a similar situation for crows. So again, out of all of these different European countries, the UK hosts the highest density of crows. So why does the UK host such high densities of these generalist predators? Well, we think there are a multitude of reasons for this, including changes in landscape composition that have occurred over the last century through agricultural intensification and afforestation. These factors have kind of changed landscape composition, altering habitats and making them more suitable for generalist predators to exploit. And also factors such as game bird release. So as I'm sure you're all aware, millions and millions of pheasants and red-legged partridges are released into the countryside every year, providing food for predators like foxes. Now, these issues and other issues like the lack of apex predators in our country are not going to be solved overnight. It's going to take changes in attitude and approach and opinion of people who run the countryside for these issues to change, which is a long-term process. Waders can't wait for this to happen. So in the meantime, conservation practitioners and wader researchers have developed a whole suite of conservation tools that can be used to restore breeding wader populations and boost their breeding productivity. Now, these tools fit into three main categories. So the first thing we can do is we can make a site more attractive to waders through habitat management and by altering hydrology, altering vegetation structure and general habitat composition. The second thing we can do is we can manage, exclude and control predators. So we can deploy tools such as electric fencing, lethal control, and even tools such as diversionary feeding. And we can also use head starting. Now, I'm sure you've all heard about Project Godwit, a fantastic project in East Anglia that is really successfully boosting the breeding productivity of our black-tailed godwit population. The thing is with head starting is that it should only be used when the population of threatened wader is at a really critical level, mainly because it costs a lot of money to head start a load of wader chicks. Now, generally, these tools have been shown to have positive impacts on wader breeding productivity, especially hatching success of wader nests. But most of these tools have only really been trialled and deployed for populations that breed in quite high densities and are quite restricted to areas in which land use is sympathetic towards biodiversity, like nature reserves. But there are also there are lots of other wader populations that breed in the wider countryside and much lower densities. And one of the species that does this quite commonly is the curlew. So in the UK, curlew breed across both upland and lowland areas. And in the lowlands in particular, they breed at low densities across a range of different land uses and different habitats as well. In the UK as well, they are declining very steeply, as Katharine said. So over the last 20 to 25 years, BBS trends suggest that we've lost nearly half of our breeding curlew in the UK, which is obviously a big loss at a national level, but it's also a big loss at an international level because the UK population makes up 27% of the global breeding population. So a huge chunk of the global breeding population breed in the UK, which makes our population of international importance. Luckily, however, there are some areas of the UK where curlew may be doing okay, and we can learn quite a lot from these areas. Now, one of these areas is Breckland on the Norfolk-Suffolk border down here. Now, these bird atlas maps show change in abundance and change in range from the first atlas period to the second atlas period. And we can see here on this left-hand graph that all these grey areas are where curlew have declined during that period, and the red areas show where curlew have increased. It's the same on this graph on the right. All these grey triangles show where curlew have been lost between these two periods, and the red areas show where curlew have been gained. So both the abundance and the range of curlew has increased in Breckland between those two time periods. So we can learn a lot from Breckland, from the Breckland curlew population, and we can understand or try and understand what conditions might be promoting this population expansion. Now, to do this and to answer this question requires some field work, which is great because that's basically the whole reason I signed up for this PhD. I absolutely love field work. But before I went out into the field, I mapped the distribution of curlew in the Brecks using BBS data BTO bird track data, and bird atlas data. So all of you citizen scientists who collected data or do collect data for those different schemes, thank you, because they've been very, very useful to me and a whole load of other PhD students starting off their career in ornithology. Very useful data, and using it, I managed to map the distribution of curlew in Breckland. Now, once I'd done this, I then went out into the field and gained access to different sites across the curlew distribution in the Brecks. Now, these sites ranged from the north of the Brecks all the way down to the south. The land use of these different sites varied quite a lot. So we've got some RAF bases here, some nature reserve, grass heathland sites, some military training areas, some clearfeld areas of forest, and some arable areas as well. And crucially, some of these sites are fenced and some of them are unfenced. So I had a nice range of breeding sites over which to study curlew. So I was ready to go out to conduct some surveys, find and monitor some nests, and do intensive monitoring of chicks as well. So what did I find out during my fieldwork? Well, before I started my PhD, the estimated number of curlew breeding in the whole of the UK lowlands was only around 150 breeding pairs. But once I had finished my surveys in 2019 and counted up how many curlews I had found during those surveys, I realised that I'd found 74 pairs of curlew breeding in Breckland. But if you remember back from the map, my study sites do not cover the whole of the curlew's distribution in Breckland. There are some really key breeding sites that I didn't manage to get access to, unfortunately. But using knowledge about those breeding sites from people that work there, I estimate that the population of curlew breeding in Breckland alone is around 150 breeding pairs. So in one field season, we managed to double the estimated number of breeding pairs in the UK lowlands, which is the first basically good news story from my PhD, and provides me with loads of curlew to study and gather data on, which is brilliant. But are these curlew in Breckland actually productive? Do they hatch chicks from their nests, and do the chicks fledge successfully? This is the next question I wanted to find out. And this required very, very intensive nest searching, especially during 2019 and a little bit during 2020. So I went out into the field and I managed to find 67 curlew nests over those 2 years. Of those, unfortunately, 43 failed. They mostly failed due to fox predation, as we can see here in this footage. Some failed due to mowing and crow predation, but only very, very small numbers. The other nests hatched successfully, which was brilliant. And this gave us a probability of a curlew nest surviving incubation of around 19%. So very, very low probability of a curlew nest surviving incubation in Breckland, unfortunately. But does this number vary? Is there any variation in this 19%? Okay, are there any sites where hatching success is a little bit higher? Well, yes, is the answer to that question. So I mentioned fence sites when I was describing my breeding sites. These sites occur on RAF bases, on nature reserves, and surrounding livestock units. They're designed mainly to keep out humans, but also to keep out mammal species such as hare, especially on RAF bases, because these fenced areas are ammunition storage areas, and they don't like hares burrowing into those areas for obvious reasons. So they're not really there for conservation purposes, but they are doing a job at keeping foxes away from curlew nests. And we can see this in our data. So this graph shows breeding probability of surviving incubation at the side, and we have unfenced and fenced areas here. We can see that the probability of surviving incubation for curlew nests laid in unfenced areas is around 16%, whereas it's a lot higher for curlew nests laid in fenced areas, around 45%. It's a similar story for curlew nests in arable fields. Now you might be quite surprised at this because this nest here looks really quite exposed to the elements, quite easy to predate. But most nests that are laid in arable fields like the sugar beet field don't really get predated. And I think this is because predators like foxes tend to stay in areas like headland areas, hedgerow areas, forest areas, and general grassland areas, rather than walking up and down sugar beet fields trying to find nests to eat. So I just don't think they encounter these curlew nests very often. And again, this shows in our data. So for grassland sites, probability of surviving incubation is very, very low. But for nests laid in arable fields, it's a little bit higher, so around 35% probability of surviving incubation for nests laid in arable fields. So could this explain why the Breckland population is expanding? Could it explain the patterns we see in our bird atlas data? Well, at the moment, I'm trying to work this out, but it's pretty hard because I don't actually have a lot of data. My first field season went very, very well in 2019, but for obvious reasons, I wasn't really able to get out into the field very much this year until the very end of the breeding season. But I did make use of this time, and I collected data on chick habitat use. During this data collection, I learned 3 quite important things about chicks and the way they use habitats. So firstly, I found out that chicks really only require around 8 hectares of grassland habitat to fledge successfully. Ooh. They also require a mix of vegetation heights and densities in which to forage and hide. So you saw chicks in that long grass, but they also use these really, really shortly grazed areas. And they also require shaded areas in which to rest in temperatures above 25 degrees. And I presume with climate change, our summers are probably going to get a lot hotter and a lot drier in coming years. So these shaded areas are likely to increase in importance. So using this admittedly quite descriptive data, we can have a look and see whether these conditions are well represented in fenced areas and in grassland areas close to arable areas in which curlew are breeding. And we can also inform habitat management in areas where curlew breeding productivity may be quite low and not many chicks are fledging. So to conclude, we know that hatching success is higher in fenced areas and potentially also in arable areas. We know that chicks are likely to need a very heterogeneous vegetation structure to fledge successfully. But overall, I do need a lot more data because I have been hampered quite badly by this COVID situation. So I need more data on chick monitoring. I need to GPS tag more adults so I can get a higher resolution data on habitat use. And of course, I need to find and monitor more nests because I wouldn't be able to go through a whole field season without doing that because I'm addicted to it, frankly. Luckily, I have another 2 field seasons left of my PhD, or definitely 1 field season, hopefully a 4th field season. I should also have some help from MSc students who will be out collecting data for me, particularly in arable areas, because they're quite hard to cover on my own. So hopefully I'm going to be getting a lot more data to answer some of these questions in more detail. Before I go, I'd just like to thank my supervisors for guiding me along the way during the first half of my PhD, all my fieldwork helpers, especially those from the BTO who helped organise access to some of my sites for me. To all my funders as well, because obviously without them, I wouldn't be able to conduct this research at all, including the BTO who part fund my PhD. And I'd also like to thank BTO in general for inviting me to speak at this conference. Now, finally, I know a lot of you like colouring reading, and quite a few of my Curlew are now colouring. So my scheme marker is a blue ring above the knee and orange white below the knee on the left leg. So if you see any of these Curlew, please report them to me. My email address is up here. Also, if you're out birding in Breckland, especially between March and July, and you see any Curlew at any sites, please let me know, and I'll be very interested. Like I said, I need all the data I can get. So you citizen scientists out there, yeah, please give me a hand. Thank you very much.
Great. Thank you very much for that excellent talk, Harry. I still always associate Curlew with the uplands in my head, so it's still quite strange to me to think that there's a whole population of Curlew, quite a large one, hiding in Breckland, only a few miles from my house. Now, we've got a few questions that have come in already, and we have the first one from Angela Spencer. She wants to know whether you can elaborate a little bit on what head starting is. Good question, Angela.
It is a very good question. So head starting is used, like I said, when a population reaches a really critical level. So when the numbers are really, really low, and on the ground conservation may not work straight away before the population ends up going extinct. And it's a technique where we remove eggs from the landscape or from the breeding site, and we head start them. So we rear them in incubation, I don't know what we call them, chambers, and we hatch the chicks, and then we rear chicks in release cages, and then when they're ready to fledge, we release them back into the wild. It's a technique that is quite new in wading conservation, but it's working to great effect. And if you look up Project Godwit, you'll be able to find out all the details you could possibly want about head starting. But it is a very, very effective tool when you're trying to boost the breeding productivity of a population that has got to a really critical level.
I think that's a really good answer, Harry, to what head starting is. We've got another one from Nori Russell, who wants to know if there's a correlation between areas that Curlew are increasing in and whether there are more basic soils present, which could be linked to better earthworm populations. I think food is something that we don't know a whole lot about for breeding Curlew, but maybe you could expand on that, Harry.
Yeah, that's absolutely right. Yes, we don't really know a huge amount about what they eat during the breeding season. But I do know that chicks, they don't eat worms. They mainly sally glean invertebrates from vegetation and from the ground, rather than penetrating the ground with their bills like adults do. I know that adults move to arable areas, or they make feeding flights to arable areas while they have a nest and while they have chicks. So the Brex is probably quite good for Curlew in that aspect, because we have a lot of arable areas in which Curlew can forage. Whether there's a correlation between soil type and areas in which Curlew are increasing, I'm not sure. I don't know the answer to that, but it could be potentially something to look into.
Thanks very much, Harry. For those who are interested in knowing more about earthworms and correlations with bird populations generally, the BTO has done some work for the What's Under Your Feet project. So I would direct you to looking up that project on the BTO's website. Now, Anne Cotton would like to know, Curlew in her area only nest in grassland and not in arable. Why would they nest in arable fields in one place, but not necessarily in another?
Well, so before I started my PhD, I didn't know Curlew nested in arable fields either. They only nest in arable fields in low density in my study area. They mainly nest in grassland areas. I don't think it's just the Brex where curlew nest in arable fields. I think they nest in arable fields in other parts of the country too. It's just that they nest there in very, very low density, so they might go undetected. Most people focus their efforts on grassland areas because that's where most of the breeding curlew are. So yeah, I don't think it's I don't think Breckland is unique in that it has breeding curlew in arable fields. I think they also breed in other areas in arable fields, but maybe less attention is given to those areas, and so not many curlew nests are found by researchers because they just don't look in arable fields very often.
Yeah, I think that's very likely the case, that there's plenty of curlew possibly hiding out there in arable fields that we don't necessarily see.
Yeah.
Now, Mike Pollard would like to know, and you might be able to tell us this from this just past breeding season, Harry, is what habitats are favored for chick rearing, and is that very different from what habitats are preferred for nesting?
So if we look at arable breeding curlew, I have seen curlew chicks hatch successfully in sugar beet fields and then be taken all the way across like a busy road, up a really steep verge, through a fence and onto a vast area of grassland. So grassland is really, really important for curlew during the chick rearing stage. If the chicks are left in sugar beet fields or in other arable fields, they're unlikely to fledge. And I haven't had any chicks fledge from a sugar beet field without them being moved into a grassland field or grassland area. So grassland is very, very important. That is also where most curlew nest. And most curlew broods do not really move very far away from the nest site, or at least in Breckland, where the habitat is quite fragmented. So the answer to your question is that nesting and chick rearing habitats are very similar.
Great, thanks very much. This is maybe a related question from Tim Arnold, who wants to know, if chicks appear to need diverse types of habitat, do you hope to get enough data to be able to see if fencing is a hindrance in terms of chick survival, presumably preventing them from moving around and accessing that habitat, even if it's a help against nest predation?
Yeah, that's a very good question. So you're right, they need quite a diverse vegetation structure, so some tall areas of grass and some shorter areas of grass. Some fenced areas in Breckland only have short areas of grass because of the management that these areas require. Like I said, some of them are ammunition storage areas. So to reduce fire risk, the MOD mow the grass very, very short, which makes predation by avian predators like buzzards and crows quite common in those areas. So potentially fences could restrict chicks getting into favourable grassland habitat. But we are working with people like the MOD to try and alter the way that they manage these fenced areas. Other fenced areas don't really have that issue because a lot of them already have this heterogeneous vegetation structure. So a lot of sites are just natural grass heathland sites that are grazed to a certain extent, which give you this heterogeneous vegetation structure. So some areas that chicks can hide in, and other areas in which chicks can forage.
Thanks very much, Harry. I think we've got time for maybe one or two more questions. So we've got one. Alan Harris would like to know whether magpies fit in anywhere as a potential predator.
So I've never had any evidence of magpies predating nests. For chicks, they could potentially predate chicks, but again, I've never recorded any evidence of that at all. And there are a lot of magpies in the Brecks, and I've done a lot of observations of curlew breeds, and I've never seen magpie predation. I've never seen curlew adults mobbing magpies either, which is often a giveaway as to what is predating chicks. So no, I don't think magpies are a big issue for curlew in general, but I'm happy to be proved wrong.
Thanks, Harry. And I think we'll have one final question, which is quite a fun one for you, I think. Martin Chadwick wants to know, where are the Breckland curlews wintering and whether you have any resightings of ringed birds?
I do have some resightings. So the most recent one was from Devon, Exminster Marshes. So I've had resighting from there. I've had other Breckland curlew have been resighted on the North Norfolk coast. I haven't had some, but some previously have been resighted in Ireland. So they go everywhere in winter. What we know is that individuals are likely to go to the same place every winter, but as a population, they go to all over the place. Basically, they can go to the north coast. Some individuals might go to the south coast. Some individuals might go to Wales. Some individuals might go to Ireland. So the migratory connectivity as a population is quite low, but with individuals, it's likely to be quite strong.
Great. Thanks very much for answering those questions, Harry. And thanks again for a brilliant talk. We're going to move on now to the third and final talk of this session, which will be presented by Colin McShane. Colin is based in Staffordshire. He's a former A-level biology teacher and has been a ringer for many, many years, and is going to tell us about some amazing technology for surveying Jack Snipe in winter. Over to you now, Colin.
Thank you. Good afternoon, everyone. And thanks for the invitation. I'm going to spend just a few minutes telling you about the work that we've been doing on trying to get a handle on the winter population of Jack Snipe in the UK, or in one particular area, using thermal imaging technology. First of all, then, some background information. I've always been pretty interested in trying new things. Way back in the day when I was teaching, trying new methods, techniques, equipment, kit, and what have you. I think probably best illustrated by something that happened in 1987. I managed to convince the area sales rep for Olympus endoscopes. They're the things that they shove in various orifices to find out how things are going. And I managed to convince him to come with me on an A-level biology trip to Lundy Island. We spent the week using them to look inside Manx shearwater burrows to see what was going on. So it was right up my street when 3 or 4 years ago, we were contacted by a wildlife-friendly keeper who was going to show us how he was using this to scan through a field which had been set aside for lapwings. And any of you that have tried finding lapwing chicks will know just how difficult it can be, how time-consuming it can be, and therefore how disruptive it can be to the breeding birds. And therefore it was absolutely amazing when the guy just walked into the field. It was a bright, cold April morning, and looked through the machine. Oh yeah, there are 4 chicks there, there are 3 chicks there, there's a nest with 4 eggs there. And within about 55 minutes, we'd ringed almost 50 lapwing chicks, which was just something that blew us away. We decided there and then that we must have one. So a bid was put forward to the West Midland Bird Club, who very generously provided us with an imager on the condition that we would do 2 things. First of all, we'd use it to explore ways in which it could be used, but not only for ringing, but for birding, for conservation uses, and that we would spend time with other organizations disseminating the information we got, which is kind of why I guess I'm here at the moment. Since then, it's been very gratifying to see that there's a small but increasing number of people who are getting thermal images and using it for various studies that they've been doing. But I think it's fair to say that certainly in my knowledge anyway, most of them have been using them for nocturnal activities, finding out a lot more about numbers of skylarks that are in fields, woodcock, grey partridges, and various other species as well. And I certainly am doing quite a lot of that myself within my ringing group. But I've, over the last 3 or 4 years, developed a fascination with using the thermal images to find Jack Snipe. And doing so in daytime rather than during the night. So we put various of our results on social media. And back in February, I got contacted by Graham Appleton. Many of you will know Graham, big in the International Way to Study group. And basically he was wanting to chat with me to see if this technology could be used to improve our knowledge of the wintering population of Jack Snipe in the UK. Now, if you look at any of the texts and the atlases, it's quoted as being 110,000 Jack Snipe wintering in the UK. So I was fascinated talking with Graham about this, because he was trying to go into how this number had been derived. There have been certain bits of information from county recorders, from county reports, from other bird reports, even some information from shooting organisations through the numbers that were in the bags that they'd been catching. But the bottom line was, nobody really had any idea. So when they came to produce the atlas, they came up with a very, very pragmatic solution, which was, let's just say there's one Jack Snipe in every one of the 110 kilometre squares in the UK, and there's our population estimate. Okay, well, I don't think we should be too critical of that, to be honest, because here is exactly the problem. This is on my prime study site, and there is a Jack Snipe only a couple of meters in front of the camera. I can't see it, and I know where it is, so I'll be amazed if any of you can see it. And of course, as many of you will know, they are cryptically colored. They blend in beautifully with this kind of background. And to make matters even worse, they don't flush, and in fact you can stand almost beside them, and in many cases they won't move or let themselves be seen. So it really is a very, very difficult task to count Jack Snipe. And this is where I think thermal imaging really can offer a solution to at least part of this problem, because this next photograph is taken 10 seconds after this in exactly the same position. Okay, now I'll grant you it's not the best picture of a Jack Snipe you've ever seen in your life, but you can see there's the head and there's the body, and you can also see the heat profile of this. It's been done on a perfect day, in that it's quite cold and dull, and therefore that gives a maximum differential between the heat of the organism, the bird in this case, and the background. It's also incredibly easy to get used to and to work out where these birds are. I had a student with me 3 or 4 weeks ago. She'd never seen a Jack Snipe in her life before. She'd never used a thermal imaging machine before, and literally within 5 minutes, she was walking around the site finding Jack Snipe as easily as I could. So it's a very, very simple piece of equipment to use. So this is my study site. In fact, it's quite interesting because it's the top of an old colliery spoil heap in South Staffordshire, and amazingly, the council, the local district council, have been preserving this and managing it specifically for Jack Snipe and common snipe. You can see they've got a little lip around the outside, and I'm standing on the lip here. Which means that the water drains into the central area. It's only about 200 meters across, almost circular. And you can see there are areas of free water there. And a closer look shows that two things. First of all, the typical vegetation there is this Juncus species, rushes. But you can also see, and this is really quite important in this site, that it's boulders strewn all over the place, and it's been puddled by heavy machinery to reduce the water drainage. And that's fine, except what it's meant is that this site is too hard for either Jack Snipe or common snipe to probe for worms and insects. So even in the areas where there's free water. So this isn't a feeding site for Jack Snipe and common snipe, but it is purely a day roosting site. Now we know that, we've been there right through the night, and there are no snipe or Jack Snipe in this area until an hour or two before dawn, and we can watch them using the thermal imaging camera coming in, settling down into the roost site, and they stay there for the rest of the day before going out again the following night. I'll come back to that particular issue in a moment. So what are we talking about in terms of numbers? Well, as you can see on the left-hand side, I try to get there a couple of times a month through the winter period. And you can see that certainly at the end of November, this is data from last year, obviously, and we had up to 41 Jack Snipe, and although I've not got it on this table, 75 common snipe, just on this relatively small site. You can see the numbers vary throughout the winter period, and but they stay quite high. And then as we get into February and March, the numbers drop off, obviously, as the birds are flying back to Scandinavia, Russia, or wherever. You'll also see at the bottom there, just as almost a side issue, I managed to ring 55 Jack Snipe on this site last year. And to put that in context, up until 3 or 4 years ago, the UK total for Jack Snipe ringing in any given year was around about 1 to 200. So it was a significant number. So, okay, so this is one site. So I've then been using the machine to test out its efficacy at finding Jack Snipe in other sites. This particular site is only about 3 kilometers away from it, and it's a much more conventional site. By that I mean it's wetland, it's boggy, the birds can feed here if they need to. However, as you can see, there's a lot more wetland grasses on here, forming quite dense clumps. But with a careful zigzag walk through this, you can still find Jack Snipe using the machine. In fact, I was there about 3 weeks ago, and we counted 35 Jack Snipe in this particular little patch here. Again, it's not very big. We've got a machine, and there are other sites where I've been using it as well. So we've got a machine and a technique which is proven to be able to find Jack Snipe if they are there. So the idea now is to derive what we're calling at this stage a holding index. And what do I mean by that? Well, the first thing is to find a large 50, 60 more daytime roost and feeding areas, as I've just shown you in the last couple of slides. And then using the thermal imager camera, count the number of Jack Snipe on all of these sites. What then we would think about doing is to decide on a range of relevant parameters relating to the habitat of each site. So how big is the site? What are the main plant types? What's the percentage cover of land? What's the percentage cover of water? Soil impaction, grazing, disturbance, there may be several others which we'll be looking at as well. And the idea is that from that, we would derive a good old algorithm. I think we used to call these a formula back in the day, to predict the numbers. So we're hoping to be able to say this site is X hectares and it's got X plants in, da-di-da-di-da, and we predict that this has a holding capacity of, say, between 5 and 15, say. So we would come up with ranges, and these are obviously would be estimates still, but at the same time, there would be at least some empirical data rather than the finger in the air, 1 per square kilometer previously. The next big stage, of course, will be scaling that up. And you can see from my slide there, that's still very much a work in progress. And when we're putting this together, I'm sitting there thinking, when on earth am I going to find time to do all of this stuff? And then, hey presto, we managed to get enough funding for an MSc student at this stage, and she started on this and going to be doing a lot of, well, all of this legwork and brain work as well. We are trying to get sufficient funding because we think that we may need more than just the 1 year that the MSc student is funded for, but we'll see, and we'll see how far we get with this. So that's basically the plan that we've got. Just a little bit about thermal images now. There are 3 players in the field in terms of providers. There's FLIR, and I've not used any FLIR equipment, so I can't pass any comment. The main player up until very recently has been Pulsar, and I think it's fair to say that the best Pulsar model is called the Helion. The problem with that is that it's between £2,500 and £3,000 to buy. And I didn't have any extra funding, so I decided recently to buy myself this one, which is in their budget range. It's called an Axion XM30, and this retails at about £1,100 at the moment. Now, it's not as good as the Helion, but it's good enough. So all of the data that you've seen, I gained using this machine. Last week I was out ringed nearly 80 skylarks in a couple of fields with it. So it's plenty good enough. The latest player in the field is HIC or Hike Visual, and they're producing models which are similar in range to the pulsar models, but they are a bit cheaper. So the model that they've got, which was the equivalent to this one, retails at about £930. But it does, in addition to what this one does, it does record, and it will live stream to a smartphone or a tablet or what have you. So there's a lot of development work going on. Prices are coming down, and I would urge you, if you're in any way interested, try and get somebody to buy you one for Christmas. So I just thought I'd show you a couple of little videos now. This is my main study site, and this is going in with a mobile phone, just scanning, and as we get in there, we then start to scan as we would with a thermal imager, round in an arc, just a few meters in front of us. And as you can see, quite easily, there are no Jack Snipe there, are there? So a few seconds later, we use the same technique with our thermal imaging camera. Now, I've not done any editing to this. It's rough and ready, but it's deliberately so, because what I want to show you is how quickly and easy it is. There's the pylons in the distance. And we scan around, and this is in real time. So in just a few seconds, tension is incredible. And there we have our first Jack Snipe. It looks like it's jumping around, but it's just the way the camera's moving. We move a bit closer and you'll see another image there. So we just move to the side because it's behind a clump of juncus, and there we have our second Jack Snipe, and then we move round to the right, and there's our third. So that's the same little area. They're all within sort of 4 or 5 meters of where we are, and it shows you just how easy these are to see with the technology. I said earlier on that I'm really keen on using technology for and new approaches to things. So very quickly before I finish, just a few other ideas for using this technology. Scanning nest boxes is so much easier with it, particularly if you've got areas where you've got ladders to carry around. You can, particularly in the early morning before the sun warms the boxes up, any boxes which have got just a black at the hole, nothing in them, anywhere it's white, is worth checking out. I'm hoping that with a friend I've got in Varanger in northern Norway, when things settle down, I hope we'll be able to go up and use it to try and find wader chicks on the tundra. It's great for looking at occupancy rates for hole nesting species like sand martins, for example. Take a photograph of the face, and any of the holes that are glowing white, they're occupied. Those that are black aren't. Got another friend that's looking at predation in ground nesting terns, and is trying out sitting in a little tent hide, using the thermal imager to study the tern colony through the night and be able to spot which predators are coming in and which are causing any problem without using external light, which of course will then affect the behavior of both the nesting terns and the predators themselves. And then lastly, I mentioned earlier on that the day roost site, the birds disappear at night, and what we're finding is unusually unexpectedly large numbers of both common and Jack Snipe in arable fields at night feeding, particularly in stubble fields, and we're actually catching both of those when we're out at night. We didn't expect that, and there's a lot more work that could be done on that. So I hope that I've sparked some interest. I hope you found the talk generally interesting, and that you'll go out and get yourself a thermal imager and join us. But before you do, I must give you a government health warning. Thermal imagery is definitely addictive and can harm relationships. We all know we're out all day birding and ringing. Now we can be out all night Way think our partners. So I hope you've enjoyed that. I'll end before going into questions with a few addresses and contacts there for both myself, our group, and the three main providers of these machines. So if there are any questions, I'll do my best to answer them. Thanks for listening.
Great. Thanks very much, Colin. That was fascinating and good to know that there's a lot of new technology open to people who are interested in trying to locate these cryptic species on the ground. I've had quite a few questions come in. The one that quite a few people are wondering is, how do you tell the difference between a jack and a common snipe on the thermal imager?
Okay, there's a really easy answer to that question. Certainly through the daytime, there won't be any jack, there won't be any common snipe there, because as soon as you go into the habitat, they quit. They fly off and they leave the jacks behind.
That was easy. Thanks very much.
And I guess it's a bit more difficult at night, but the commons are distinctly bigger than jacks. But in the daytime, and that's why when I sort of joked about it's not a very good picture of a Jack Snipe on the first image through the imager, but everything else will go. So any passerines, reed buntings, meadowpipits, skylarks, they'll move out of the site when you go in and start walking around. And basically the only things that are left are Jack Snipe.
Great, thanks very much. And for anybody who wants to know how to identify jack versus common snipe in the field during the daytime, if you see a bird versus a white blob on the thermal imager, we have a very helpful identification video. So head over to the BTO identification video series on YouTube to find that out. We have a couple of questions here. We have one from Stephen Chapman, who wants to know what the range is for detection with the thermal imager.
Okay, that depends on two things. First thing, it depends on the quality and the specification of the machine that you've got. So I did say that the Helion is better, and that's one of the ways in which it is better. It's got a better long distance range. The second thing, however, is the type of vegetation. So in other words, if you were looking on a field that was ploughed and tilled and was flat, you could see skylarks from 50, 60, 100 meters away. But if there's dense grassland on there, or if there's a crop growing, then it cuts it down to maybe 20 meters, depending on the density of the vegetation.
Great, thanks very much, Colin. We've got a few people wondering how you can catch Jack Snipe if you want to ring them.
Okay, well, on that particular site that I use as my main study site, I use the machine, find the Jack Snipe in the machine, then find it with the naked eye, and I've got a hand net that I just put over the Jack Snipe. We have had one session on that site where we did use conventional mist netting, and we ended up we caught 16 that night, which was an incredible catch. But it's an exposed site, so there's not that many occasions when the weather is just right for us to do that. But it's basically a hand net and sneaking up on them. Unfortunately, they do tend to sit.
Great, that's very helpful then that they stay put when you try and approach them with a hand net. We've got one person who's asking about whether thermal imaging would work for curlew, and this was actually something we were talking about the other day. Yeah, so maybe you can answer that. I'm sure a lot of people might be wondering.
Yeah, I mean, the difference is, of course, well, there are many differences, but one of the key differences is that you'd be looking during the summertime, or at least the springtime, for curlew nesting. I assume that's what the question is. And therefore, what happens is, particularly if you've got a sunny day, the vegetation really soaks the sun up and heats very, very quickly. So you end up looking at a lot of white blobs, i.e. the clumps of grass, and you're looking for a white blob, which will be the bird, in the middle of that. Now, the thing that might make it easier, and this is all off the top of my head because I've never actually tried this, but one of the differences is that curlew are quite big, and if they stand with their head above the grass line, you would be able to see them. But having listened to the Katharine's talk, when the sitting bird hunkers down on the nest, it might be much more difficult to see them. So the answer is I don't really know, but I think it's got to be worth a try, but I think it could be quite tricky.
Great, thanks very much for that, Colin. We've got one question from Jane Puzzy, who wonders, I just want to check whether common snipe and Jack snipe are crepuscular nocturnal in terms of their activities.
I think they're both. So in other words, if you go out birding to my local site is Belvide Reservoir, you go out there, you can see common snipe and Jack snipe feeding during the day around the margins of the lake. But also we are finding them feeding at night in stubble fields. So I think both is the answer.
Great, thanks very much, Colin. And I think we'll probably do one more now. So Bill wants to know what your estimate of the UK Jack snipe population might be as a result of this work. And obviously that might be quite difficult to extrapolate. So maybe for your particular area, you might like to address in terms of your population estimate and how this method might help get a better handle on that population estimate for the UK as a whole. Yeah,
I just don't want the answer to be 110,000. No, seriously, it's really early days. I spent the previous 2.5 or 3 years practicing catching them on this site, practicing aging them. They're really quite tricky species. And it wasn't until February this year when Graham contacted me about this notion. And really, we've got our research work starting literally as we speak. She's going through the first stages of that. So it really is difficult. My hunch is that if I can find sort of 70 or 80 Jack snipe on 2 small sites in South Staffordshire, 2 kilometres apart, it seems to suggest to me that 110,000 is probably an underestimate. Having said which, that population isn't a static one. From the stuff I'm finding, birds are moving through. We're catching very few retraps on our site. So there's a whole lot of work to do yet on where birds are going and how what the site fidelity is. So I can't really be any more specific than that, I'm afraid. Sorry.
That's all right. Thanks very much, Colin. I think we'll wrap it up there now. I think we've got most of the questions now. So thanks very much to all the speakers for offering a fantastic session of free talks. So thank you again for your fascinating contributions, and to all our listeners out there for joining in our virtual conference. As I mentioned at the start, now more than ever, BTO's research relies on your support because of the impact of the COVID-19 pandemic. And there are many ways to support us, and many of you already do great things for us. However, if you're able to support us now with a donation, then we'd be very, very grateful indeed. And you can do that by following the link that's on your screen now, www.bto.org/support. I hope that many of you will be able to join us for more talks throughout the week. Our next session will be taking place tomorrow morning from 10 until 11:30, when we'll be hearing about understanding bird declines. First, about the fantastic joint partnership project that is Red 67, and then on BTO's fascinating research on Arctic skuas, and finally, migration and feeding movements of puffins. So until next time, thank you very much again, and enjoy the rest of your day.